organized under the aegis of DBT star status. I would request Dr. Sejal Rathod, Head Department of Microbiology and Coordinator of Biotechnology, to highlight the theme of this webinar.
Thank you, Prachi. So very good evening to everyone and good morning to Dr. Tejeshwar. On behalf of KC College, Departments of Biotechnology and Microbiology, I welcome all of you all, and especially our alumni speaker, Dr. Tejeshwar Rao, colleagues, teacher participants, student participants on this webinar on growth factor receptors. This webinar that we have organized is a part of our Alumni Connect series, where we invite our eminent alumni to share with us their journey that they have taken after graduating from KC College. Tejeshwar has been our alumni from the Department of Biotechnology, batch of 2008. The purpose of having this webinar is multifold. Not only we get to connect with our alumni, but the knowledge that we gain from the cutting edge research that they are carrying out. It will also inspire our students who are currently pursuing their graduation and post-graduation at KC and other institutions. So I look forward to this webinar. I think it's going to be enriching and engaging one. Welcome everyone once again. Yeah, thank you. Over to you, Prachi.
Thank you, ma'am. I also take immense pride in introducing to you our principal, Dr. Himlata K. Bagla, who is the head of Nuclear Radiochemistry Department and Senior Dean of Science and Technology, HSMC University, Mumbai. I would like to thank ma'am for always being a guiding light at the end of the tunnel. I now take this opportunity to introduce to you our speaker for the day and our alumnus, Dr. Tejeshwar Rao, who has completed his PhD in Cell Developmental and Integrative Biology from University of Alabama, USA. He has completed his Bachelor's in Science, Bachelor's in Biotechnology from KC College in the year 2008. He's currently working as a postdoctoral associate on imaging the cellular dynamics and mechanical forces generated during cell adhesion and receptor-mediated endocytosis at the University of Alabama under the guidance of Dr. Alexa. He has also worked as a teaching assistant for applications of microscopy at University of Alabama and a graduate teaching assistant at Wayne State University, wherein he has taught various courses like neurobiology, anatomy and physiology, comparative physiology, and basic life mechanisms. He has been invited as a speaker at various research seminars and several publications under his name in various international journals such as Frontiers in Cell and Developmental Biology and Journal of Cell Biology. To add another feather in his cap, one of his research work was recognized and selected as a cover image by the Journal of Cell Biology. Dr. Rao has won several presentations of prestigious awards and honors, to name a few: Best Poster Award in Cell Mechanics, Mechano Sensing and Motility at Biophysical Society 63rd Annual Meeting at Baltimore, Maryland. First place in both platform and bliss presentation at the 16th Annual Postdoctoral Research Day and Graduate Teaching Assisting Award at Wayne State University. Thank you so much, sir, for making it possible to have you amongst us today. I am sure our participants are eager to hear you. Kindly address the audience. Thank you.
Thank you so much for this wonderful introduction. I hope you guys can hear me. I am totally overwhelmed and nervous for multiple reasons. One being I am myself a former student of KC College. I was in Department of Biotechnology. And second, I'm sure my entire family is watching me present right now. And this is the first time they're actually listening to me present a scientific talk. But before getting into the details of the science, I have to thank the organizers, especially Dr. Sejal Rathod, for giving me this opportunity to present my work. We completely understand that given the situation, it's extremely difficult to organize such an event, and kudos to the entire team for making this international presentation possible. As you can see from the title of my slide, my research involves studying these unique structures formed in the cell. I call them cellular fireworks because it looks like they are emanating from the center of the cell. But these are the mechanical forces a cell generates in order to attach and spread on any substrate. Today, I'm going to specifically talk about one of these growth factor receptors called the epidermal growth factor receptor and how it acts as a mechano-organizer in regulating cell spreading and cellular tension. Before going into the details of the science, I thought since it's more of an undergraduate audience, I should share my research journey with you guys to portray how diverse and different it has been. So when I first started, my first research opportunity came at Casey College, all thanks to the Science Honors Program. And I had an excellent mentor in Dr. Anupama Herschel, who actually helped me through my project. And back then, my project involved studying bioremediation of wastewater using enzymes from like vegetables and then combining it with fly ash. And I thought I was really interested in studying environmental remediation and biochemistry. Keeping in line with it, I went on to do my master's from Institute of Science, again, one of the institutes in Churchgate, where I met an excellent mentor, a person who totally changed my perception of science, Dr. Suresh Mahajan. Unfortunately, he's not with us anymore. However, there I was exposed to doing science at the Baba Atomic Research Center, where I studied purification of Bacillus sphaericus binary toxin proteins. And that got me interested more in proteomics and biochemistry. But then when I started doing PhD and I was enrolled in a PhD program at ACTREC Kargar, I started studying cancer. So I kept changing fields right through my bachelor's to my master's to PhD. And I thought, okay, cancer is going to be the thing that I really get interested in. I would like to mention Dr. Manoj Ramteke at ACTREC, and he was actually my colleague, but a senior colleague who taught me how to think as a student in research. And that helped me improve my perception of science and my approach. But then I did not continue long with my PhD in India. I thought I would like explore opportunities abroad. So what I ended up doing was I applied for my PhD and I went to Wayne State University at Michigan. And there, under the guidance of Dr. Arun Anantharam, I was first introduced to microscopy. And most of my talk is heavily microscopy based, but I'll try to make sure that you guys understand and I break it down. I would also point it out that my PhD project was neurobiology based because I studied secretory granule heterogeneity and how secretion is regulated in a calcium triggered manner. So again, as I went into doing my PhD, I changed my fields. And this was nothing which I had either studied or been exposed to before. Once I got done with my PhD, I joined Alexa's group and I continued to work with them. And I changed my fields again. I started studying mechanobiology and cellular tension, trying to develop new cutting edge microscopy techniques in order to facilitate research in biology. So why do I find it so important that I put a message across that I kept changing my field? The reason is science is a very interdisciplinary field, and you cannot really concentrate on one subject or one topic and hope to achieve progress. As undergraduates, my sincere advice to everyone who's present today is that keep your minds open, take every opportunity possible, and always be flexible. As you can see here, I've put together a chord diagram representing the affinities between different research areas. And what you can see is that none of the fields can function by itself. There's always an interconnection between different subjects. And the more you expose yourself, the more flexibility it gives you going forward and developing your own skill set. And in doing so, you would come out with excellent research. So that would be the first message of my talk that I want you undergrads to take with you going forward. With that in mind, let's go look at what I really do in my thesis lab. I am interested in studying how cells interact with each other and their environment. What do I mean by that? So, cells, if we consider these two cells, they hold on to each other by forming something called junctions. Specifically, these junctions are known as desmosomes. Now, you have to understand it's like shaking hands. If you want to shake hands, you want to exert some force. If you are unable to do that, you wouldn't be able to gauge the sensation of shaking hands. Likewise, when cells hold on to each other, they experience mechanical resistance, and they could sustain this mechanical resistance by forming junctions called desmosomes. That is one of the topics which I wouldn't be presenting today on, but I also study this other type of junction, which is between the cell and the extracellular matrix. So cells do not always lie in a free space. They are not always free floating. They are attached to some form of basement membrane or some 3D matrix. Something like your skin cells are attached to an underlying 3D matrix, which involves flesh in raw terms or like the underlying bone. And they do that by forming focal adhesions. And that is something that I will be talking about today. Again, you don't have to remember these terms right now. I'll try to break it down as I go ahead and make it as simple as I can so that everyone can follow. The third topic that I study and I've not highlighted is, let me bring my laser pointer in. Yes. Are these small small intercellular vesicles called, can you guys see my pointer? I'm going to assume you guys can see my pointer. So there are these three small vesicles which get endocytosed, and I am interested in studying triggered endocytosis and developing tools for it. So when you start looking at the scientific aspect of it, specifically, you would see that the three aspects that I just described can only be studied if we start developing microscopy tools, because each and every junction is in the nanoscale, and you need extremely high resolution to look at details. And these are some of the images and pictures that I've taken in the lab, of course, with help with other colleagues in the lab. And I thought it should give you a flavor, at least a visual flavor, to understand the diversity that a cell could possess. With this in mind, I will give you an outline of the talk, which is mostly going to be based on the cell adhesion mechanics. So I'm going to start by giving you a background where I hope to convince you why is it important to study mechanical forces, and specifically highlight what is EGFR integrin crosstalk, because I assume you guys might not even be aware of these proteins. Or if you are, you might not know the mechanical role these proteins play in our bodies. I'm going to explain the approach because studying these proteins in isolation is extremely difficult. So we had to come up with unique probe designs and experiments, and also strategize in terms of our microscopy approach to better understand the question at hand. Finally, I'm going to show you some results and hopefully make sense out of them, and conclude by showing what we think is happening within our cells and what we think is a novel and important contribution to this field. So let us first understand why is it important to study mechanical forces. So as you can see, mechanical forces dictate any and every process within the body. It could be as simple as when you have some kind of tissue damage, if you get hurt, if you have a skin blister, and then the tissue or the skin has to regenerate, it would involve rearrangement of cells, which is a mechanical process. Likewise, if there is development of blood vessels, organs, growth, all these cells need to actually generate and reform, and in doing so, develop these 3D structures, which then can perform normal functions. Another aspect where cellular forces play a very strong role is metastasis, which when one of the mechanisms by which you get cancer and spread of it. So it's very important both to study regular functioning and irregular functioning in the body. And studying mechanical forces could give you some insight into what strategies could be used to either develop tools to remediate any abnormality, or to strategize to improve what goes on in a normal environment. Other examples would be, as I suggested before, a cell-cell adhesion, which would be a cell-to-cell contact, something which could be between two cells, cell and the matrix, between the nucleus and the underlying matrix. And there are multiple different options within a cell where forces play a vital role. So the take-home message from this slide would be mechanical forces guide diverse biological processes, and cell mechanics are misregulated in different cancers. And I would like to highlight and point out that it's not just cancer, but many different diseases which are strictly regulated by the constraints of mechanical forces. So the question would come out, since I am interested in cell matrix adhesion, I am going to specifically try to explain to you guys how are mechanical forces generated during cell matrix adhesion. I always like to start by giving this example of a cell. So this is one of the cells I had imaged. I've stained for focal adhesions. So what are focal adhesions? These are contact points which are formed between the cell and the extracellular matrix. And I've highlighted one such focal adhesion in this expanded 3D cartoon. As you can see, it's composed of multiple different proteins. For the purpose of this talk, all you have to remember is this anchor protein called integrin. And integrin happens to be a transmembrane protein. So it would actually pass through the cell membrane and then anchor on to the extracellular matrix via multiple different mechanisms. As long as you can remember that, we are golden. We don't need to know of all the other proteins around. But what is important to know is this complex structure then holds on to the cell cytoskeleton, and in doing so, helps the cell define its shape, helps it maintain its shape, helps it to change its shape when it wants to migrate, or it wants to proliferate or differentiate or grow. What is also interesting to know for the purposes of the talk is that when this protein, as I've highlighted here, integrin, interacts with the extracellular matrix, there is an outside-in signaling. What I mean by that is, when this protein interacts with the environment, there is a cue or a mechanical signal sent into the cell, where the cell now knows that it has formed an anchor point. And once it gets that signal, it sends a reverse signal outside, because the cell wants to communicate with the extracellular matrix. So it's a bidirectional communication which goes on. Now, if you start making this model more complex, one of the primary things you need to add to this model are growth factor receptors. And the reason for adding that growth factor receptor is your cells don't have isolated proteins. Cell membrane has more than 3,000 proteins floating around. And easily proteins crosstalk, okay? They communicate with each other. So imagine the cell membrane to be the local train in India, where you have multiple different personalities and characters all mingling together. And there would be some crosstalk, you know. So you cannot have a signaling mechanism in isolation. It has to be a coherent function. So what these growth factors do is they go and poke or interact with integrins. And in doing so, they could modulate both the inside out signaling that I was talking about and the outside in signaling that might be involved. So if that is true, and we know that integrins are the primary force generators, the question we like to ask is, does this interaction in some way or form modulate the force being experienced by the cell? Um, if I had to just break the suspense, the answer is yes, because this is not something which I have established. Lots of people have shown that any interaction between integrin and other signaling molecules leads to an increased force output. And this changes based on the state of the cell or the disease of the cell. But what is not known is what is the exact mechanism. Is it a one-to-one interaction? Is it a direct interaction? Is there a direct force change? And those are the aspects that we are really interested in answering. I would like to convince you a little more why it's important to study this interaction. So when you look at integrin, like I said before, the anchor protein, which is a transmembrane protein, I have mentioned previously that it interacts with the microenvironment. And when it does so, it regulates all these multiple functions. It could regulate the cell shape. It could regulate how fast the cell moves. It could regulate whether a cell survives or dies. Going through a cell cycle, gene expression, every aspect that I've mentioned here is directly regulated by integrin matrix interaction. What is interesting is all these aspects are also regulated by EGFR signaling, which is epidermal growth factor receptor signaling. Now, if two proteins on the plasma membrane both dictate same functions, it's only logical to think that they might be regulating these functions, interacting with one another. And it's not an isolated case where one protein does one function and the other protein does not touch that function at all. So a cell being a complicated system, all functioning is regulated by crosstalk. And that is what we are interested in exploring. Another reason to explore this crosstalk is any abnormality in signaling would lead to a disease. And there are multiple diseases associated with either the integrin signaling defects or the EGFR signaling defects. What is interesting again is all these defects are common. It could be heart-related disorders. Cancer is one of the most common ones. It could be rheumatoid arthritis, Alzheimer's, you name it. And you will find mutations or abnormalities in both these protein signaling in that disease. So that motivated us to study the crosstalk. And the other important thing missing in this field is whenever they study this crosstalk or they try to study this crosstalk, they do it significantly downstream from the membrane. What I mean is these are two proteins which are on the cell membrane. So whatever action is happening or whatever changes are happening are at the cell membrane. However, there aren't many clear techniques available to study the interaction at the plasma membrane. And I will tell you why in a little bit. So all the literature that is available in terms of this crosstalk is by people doing biochemical assays or ELISA-based assays or any assay which is based on intracellular signaling. And as interesting as that is, it is not hitting the nail at the head. Okay, the main point is this interaction happens at the membrane. So we need to study this at the membrane, whether there is a direct physical interaction, and how does this interaction influence the inside out signaling. And those are the aspects that I intend to cover in my research today. Um, Another point, just to reiterate what I said, these are some of the papers published in this field. And don't worry about the words on these papers, and don't even worry about what I'm showing. The only message you need to take is when they design these models, all that they've highlighted are signaling mechanisms deep inside the cell. There is no mention of what is happening at the membrane or outside the membrane. So this lack of information is vital, because if we could get that information, we could use it to design or come up with drugs, remediation strategies, trying to develop sensing platforms, etc. Okay. Now, if this interaction is so important, why haven't people studied it, right? Like, that's the question. Like, I keep saying you have to study it at the membrane and outside. Why do people just not study that? The reason is it's very difficult to study these proteins at the plasma membrane. One of the reasons, which I stated earlier, the membrane is so crowded with so many different proteins that you cannot study single protein expression and signaling in isolation. Two, even if you could, you cannot parse out the contribution of one signaling pathway from the other. So you really need a clean system, an isolated system, to study these signaling mechanisms separately from one another. So we were really troubled, and we thought, how could we address this issue? So we came up with a unique design strategy, and I'm going to spend a lot of time trying to explain this design strategy, because if you can take this from the talk, I would consider my talk decently successful. So we said, okay, we want to study membrane proteins and we want to study them in isolation. So why not design a glass surface based strategy? So what I do is I take a glass cover slip and then I functionalize the surface. You all study chemistry, and I'm absolutely sure you've studied surface functionalization. It's as simple as forming a streptavidin biotin bond. Okay, that is just my base bond to attach my probe, which is actually a DNA based probe. As you can see, it's a double-stranded DNA. Don't worry about the sequence. What is important to note is I have somehow managed to modify this probe to have a fluorophore and a quencher. What is a fluorophore? Something which fluoresces. Okay, it's going to shine. What is a quencher? Something which is going to stop it from shining. And then if I can manage to make this structure, I could put any functional group I want at the end of the probe. And then if my protein reacts or likes that functional group, I could study the interaction of any protein in isolation from everything else. So with this design strategy in mind, we thought that if we could make this surface, and this is my cartoon cell, if I could put my cell on the surface and I've enlarged this box, you can see if integrin were to react with my ligand, then it would open the probe because it's going to try to pull the double-stranded DNA apart. And if it pulls it apart, I would have a signal in terms of brightened fluorescence. Okay, it's going to shine brightly. So every time the cell pulls on my probe, I'm going to get a signal. So we came up with a visual strategy using DNA-based sensors to visualize forces in real time. Now, another thing which I said was these are tension gauge tethers. What does tension gauge tether mean? Okay, it's going to report on the actual tension being experienced at that molecule. How does it do that? And why is it important? Before telling you how it does that, let me tell you why it is important. So if you have ever read any scientific paper, um, I would like to inform you that if someone publishes a paper in Science, it's considered a rule of thumb that that is the ground truth and it's the most innovative thing ever. So we have this paper from one of the pioneers in the field, TJ Ha, who showed that based on the tension experienced by a cell, the spreading behavior changes. And there is a cutoff threshold under which cells do not spread. They don't even attach. So in this image, if you can see, they have tested different tension thresholds, and they see that when the tension threshold is below 33 piconewton, and understand it's piconewton, so it's 10 to the power minus 12, okay? Does not matter. All you need to remember, it's extremely, extremely small. So if a cell cannot experience that force, it would not attach and it would not spread. And if it does not do that, it's not going to grow. So this is the main reason we came up with a design where we could sense the force, because we wanted to know if these values were actually correct and if we could somehow change those values and change the cellular behavior, because that is what happens in a diseased state. But for that, we would need different reporting strategies, right? We need to not only know what the force being generated, but if it is more or less, what is the number value associated with that force. So we went back to our design and we came up with these two different strategies, okay? I know they both look same, and it almost look like they've been flipped, but that flippage creates a big difference in its detection limit. What should be noted is the location of the fluorophore and quencher with respect to the point that it is attached to the substrate, okay? So there is one conformation, which is the low tension conformation or unzipping conformation. What I mean by that is it's like how a zipper works. When you try to open any zipper, initially you experience some force, okay? But that force is not tremendously high. It's very low. Once you've started opening the zipper just comes apart easily. What that means is if there were a double-stranded DNA where the fluorophore and quencher are away from the actual stem where it is attached, once the probe starts to open, it would be ruptured very easily. On the contrary, if there was a fluorophore and a quencher which was attached to the site of the stem, you would only see a signal when all of these bonds are broken, correct? And that is the strategy we employed. Also, what we did was we played with the ATGC sequence that we use for this DNA double strands. So we made this AT rich, since A and T form only double bonds. So double bonds are easy to break. And then we made the shearing mode GC rich. So it takes more effort for the cell to pull that probe apart. So by having these two different flavors, now we can gauge two different tensions being experienced by the cell. The take-home message from this is now, not only can we get a numerical value to the force being experienced by the cell, but we have the ability to gauge the two ends or have a big dynamic range to gauge the two possibilities where a cell might not like it versus a cell might like it. With this in mind, we thought, okay, now we have a tool, but can we still use it? The answer is yes, if we can somehow put it on a microscope and start looking at these forces. So to remind you again, I'm going to make this platform on a glass surface, which is going to be DNA based. Then I have to put my cells on that surface. And when my cells start to pull on the probe, I could put this entire glass cover slip on a microscope and then look at my cells in real time. But then there is a limitation of the microscope. And what is that? So generally, when you image cells, and I think you guys might have imaged or at least looked at something under a microscope, what happens is the incident light passes straight through your sample. Now that is good for general purposes, but very bad for fluorescence imaging. And why do I say that? When light passes straight through a sample, you see everything that is illuminated. So let's say there was a fluorophore molecule at the cover slip cell interface, or there was a fluorophore molecule inside. Everything is illuminated, everything is shining, too much background, too much of out of focus fluorescence. How can we modify it? We play a small trick where instead of passing the incident light straight through, we bend it. So we bend it at an angle. It's called a critical angle. And in doing so, rather than the light passing straight through the sample, it's totally reflected internally through the objective. And then it only fluoresces molecules that are really close to the plasma membrane surface. So if I can do that, then now my resolution improves tremendously. And instead of looking at all this garbage in the cell, I can start looking at individual structures inside the cell. And that would help me understand the specifics and the intricacies which I intend to study in terms of the forces being generated. Because understand, these are single molecule forces. This is a single protein molecule which is generating a force. And if I had this out of focus fluorescence, I am going to be confused as to where it is coming from. Is it really coming from the membrane? Is it coming from inside the cell? Is it even the cell that is generating the force? Who knows? So by making this small modification in microscopy, we could actually improve and help use the probe that I just described. At this point, I would also take a break from the science and advocate for studying microscopy. And this is again because you guys are young and you need to understand that microscopy is the future. And in order to highlight that, I've put all these STEM education logos from different websites. So basically, you see, whenever they talk about STEM education, science, technology, engineering, and math, they always put the microscope symbol on the STEM logos. And that's because microscopy is absolutely essential to study anything in the scale from, let's say, something as big as a whale, but you want to understand what's going inside the whale, to something which is like a single molecule, which is what I'm studying. And based on the type of microscope you're using, you can you have different dynamic range or different sizing that you can see. So my advocacy right now is that if you get an opportunity, try to understand the microscope you're using. And if you have more opportunities, try to learn the different kinds of microscopes or techniques available, because it would help you in your scientific career, irrespective of the path you decide to choose. Okay, with that in mind, let's go back to the science. So we started asking questions. Okay, we have a probe, we have the microscope. So let's start asking scientific questions. The first question we asked is, as I told before, EGFR is an epidermal growth factor receptor. And the way a receptor functions is by binding to a ligand. So the first question was, we see these cell mechanical changes, right? We know theoretically there should be a mechanical change. Is it ligand dependent or is it ligand independent? So I'm going to go through slowly on my images because I again know this is very, very complicated, but I'm going to break it down, okay? So this is one of those cells which I have put on the tension surface. And the first thing I do is I want to make sure the cell has spread. So I take this first image, which is called RICM, which is Doesn't matter. You don't need to know what RICM stands for. All you need to know, it is a technique which allows you to look at the cell footprint, okay, or the cell imprint. So it exactly tells me the spread area of the cell. And then I start looking at the fluorescence aspect. I consider myself a fluorescence microscopist, and I love seeing things glow. So as I told before, my tension probe lights up when a cell generates tension, and that is recorded in this channel. Now, it is also important that I look at the underlying structure. And this is one of the proteins, which is part of that complex. You remember I said it's a very complicated complex called focal adhesion. So this is to mark that complex such that I can then correlate my tension with my focal adhesion marker. Okay. And then I used another marker, which is for looking at the cell cytoskeleton. I want detailed structural intricacies, and I can see that the cell is complicated. And then I can overlay and say, look, force is generated at the same site at focal adhesion formation. It helps cytoskeletal organization, etc. Now, this is on my stronger threshold tension surface. What is important to point out here is I have my ligand present. So I have written plus EGF. But if I take my ligand away, the cells hate it. They don't want to spread. They don't generate tension. You can see this radial, beautiful pattern with a round distribution. That distribution is lost. The paxillin organization is lost. Cytoskeleton is not polymerized, so it's not making the cells spread, and the cell looks really, really unhappy. So just by adding the ligand for EGFR, I can change cell spreading and cell tension, which means these two things can regulate cell mechanics. I did the same experiment on the 12 piconewton, which is the softer substrate. And we found something really interesting, something totally different from what this field has previously suggested. We found that in presence of the ligand, cells start to spread. So if we brought in the growth factor signaling into the whole scheme, cells now start spreading. They start generating tension. They start orienting their focal adhesions radially, and then they have actin polymerization. Now, this is novel because if you remember the previous thing I showed, previous concept or terms in this field is that cells only spread on higher threshold surfaces. Lower threshold surfaces do not allow cells to spread. But what all this field has missed for the past 20 years or so is that they do all these experiments without looking at growth factors. They do it in media without serum. And when you don't have serum in your experiments, you do not have growth factors. And if you don't have growth factors, you are making your cell unhappy. It's just like if you don't eat, you're going to feel weak. Likewise, if you don't provide growth factors, your cells are going to be weak. They are not going to be happy. And then you're looking and studying unhappy cells and trying to understand biology is not a good scientific approach. So we alleviated that with this isolated system approach. And as you guys know, any science is only valid if you can perform statistics on it. I can try to convince you showing these images, but no one would believe me in the scientific community. So we always quantify these. And what we quantify is the total tension that is being generated, which has been depicted here in the integrated intensity graph, and then the RICM area, which is the total cell spread area. Again, you don't have to understand the details of this quantification, but all you have to take is we have statistics to back our claim that indeed ligand stimulation leads to enhanced cellular mechanics. And it is ligand stimulation that dictates integrin tension and cell spreading. With that in mind, we got very excited. We said, okay, let's go and see more about what is it about EGFR that regulates focal adhesion organization. So we zoomed into these images and we saw these beautiful patterns, like really radial distribution. and we wanted to quantify it and, make sure that indeed every streak that you see with the tension co-localizes with the paxillin. And we did some co-localization analysis, and indeed we see perfect co-localization. You see really high amounts of co-localization with EGF, and when we take EGF away, the co-localization falls apart. Likewise, we also studied other things in terms of like looking at the focal adhesion size, where I actually go in and measure this size and understand these focal adhesions are micron size. So they're really, really small structures. But also we are using a really high-end microscope, which allows us to like totally expand things and like get nanometer resolution. So we could make those measurements, and we saw that indeed activated EGFR regulates the maturation process of focal adhesion. So with these quantifications, we could start making claims that indeed, when we stimulate it with a growth factor, the growth factor receptor acts as a mechano organizer and brings about these dynamic changes, which was previously not explored at the cell surface. These are evidence at the cell surface. These were fixed images. I did all these experiments by fixing and staining. Now, the scientific field is very critical. They like to see things live. They like to see things change actively. So we did an active experiment looking at EGF stimulation dependent changes over time, where I am constantly imaging for 90 minutes, and I could look at the cell every minute as it changes, and it starts exploring the environment. It starts giving out this tension profile. And what was interesting was I did it in 2 different flavors. One, for a long period of time, for about 40 minutes, I would keep the cells in media without the ligand. And then suddenly I would switch it to having the ligand. And I hope you can appreciate that the change in tension happens at the point of me stimulating this whole cell. And it indeed tells us that, the growth factor stimulation is absolutely essential for cells to spread and generate tension. And then we played a cruel trick on the cell, where instead of not having EGF and then having it, we gave it EGF, but then we washed it away, kind of to make sure and understand if it's only required for growth or if it is required for maintenance. We were trying to tease apart the different stages at which the growth factor might be required. And this gave us a result which showed us that once the cell has spread, once the cell has generated tension, it does not fall apart. It can maintain that state even if we take the growth factor away. So as the term might suggest, growth factor is required for active growth. And once that active growth state is attained or is plateaued, the cell can sustain for smaller amounts of time without having a boost of that growth factor. So that helps us understand that, this initial tension spreading is a direct outcome of presence of ligand. Now, everything is fine. When you start adding ligands, things change, but is it truly receptor dependent? Is it truly a cytoskeletal phenomenon? So whenever these kinds of questions come in the scientific field, we try to hit the cells with some kind of inhibitor, which is a drug. So just perform the same experiment, exact same conditions, but now put something which would inhibit some protein which has been shown to be extremely important. In our case, we went for the cytoskeleton. Why? Because we know if a cell has to spread, it has to polymerize its actin. And in doing so, the cells spread. So let's take all the actin away. Let's try to break that backbone such that the cells fall apart. A parallel example would be our own skeleton supports our body, right? However, if I were to put some chemical in our body where I would break down all the skeleton into smaller pieces, our body would fall apart. That's exactly what I'm doing here, where latrenculin is an inhibitor of actin. and blebistatin is an inhibitor of myosin. And you see almost near complete loss of tension. The tension goes away, the organization goes away, the polymerization goes away, totally destroys it. Did this experiment both in presence and absence of EGF. And whenever I hit it with the drug, cells hate it. They don't like it. And that's normal because I'm being really cruel to the cell. Likewise, we quantified it because, again, quantification is of vital importance. And we see that our measurements depict our visual observation. So we can finally say that EGF stimulation by itself cannot override the effects of cytoskeletal inhibition. Because if that was true, when we were stimulating with EGF, we should have seen maintenance, and we don't see that. So we make these claims, and that's more for the scientific community to for future research. We also went ahead and used inhibitors against EGFR because we claim, right, like everything is happening through EGFR, but who knows? There are so many proteins on the cell membrane. I might be making all this thing up. So we used inhibitors against EGFR, 2 of them. And we chose this because a little known fact, these are also the inhibitors which are used for cancer treatment. So there is a lot of EGFR overexpression associated with lung cancer, breast cancer. And when you go to the hospital or you go to the doctor, they would prescribe you erlotinib hydrochloride. That's one of the most popular drug given for lung or breast cancer treatment. It sells under the name of Tarceva. And we show that using this erlotinib, we can actually inhibit the tension and the cytoskeletal profile. And it looks dark because everything is destroyed. So it's a normal image. It's not an image manipulation. It's just a showing that, again, it is EGFR driven. So with this in mind, we quantified it. Again, not important, but what you should take or remember is that these inhibitors are working through EGFR, which tells you that EGFR is indeed the kinase domain of EGFR. As you may or may not know, the receptor is a tyrosine kinase domain, and it basically phosphorylates other downstream proteins and brings about all its functionality. And inhibiting that kinase domain with either erlotinib or tyrosine totally inhibits integrin tension and spreading. So that should tell you that EGFR signaling is absolutely essential for these processes. Now, people in the scientific field would not be convinced if I used an inhibitor. And I will tell you why, because inhibitors are brute force chemicals, and they just totally hurt more things than they say they do. So as specific as they can be, they're still non-specific. So we adopted a more specific approach, which is a silencing-based approach. We used siRNA or silencing RNA, which would go and inhibit the protein itself. And siRNAs are very, very specific. So we targeted EGFR. And we see that when we use this, we did control experiments, but that's not important. What's important is when we use siRNA against EGFR, we see a near complete loss of tension, even in presence of EGF, and we see near complete loss of all these cellular structures. So this is proof of principle. What we show with inhibitor, we validate with another experiment. And that is something, as a young scientist, you guys should keep in mind. You cannot show a result with only one method. You always have to show that it works irrespective of the method, and then you can make claims. And that is the accurate scientific approach I would suggest to follow in you guys' career. With that in mind, we again quantified it, and we can confidently say that EGFR silencing causes significant loss of integrin and cell spreading. Now, since everything is happening with EGFR, we were interested in knowing what is it about EGFR, or what aspect or what portion or what functioning part of EGFR regulates the tension outcome. So I put the slide here because many of y'all might not know how EGFR functions. So to give you a brief description, EGFR is a single transmembrane protein, and it has this domain that I've marked called a kinase domain. Okay. And what happens is when the ligand goes and binds to the protein, then single molecules come together and they form a dimer, and this dimer is active. That's why I'm showing that all these phosphorylations on the protein. Now, once the kinase domain is active, it could recruit downstream signaling molecules and pass on the signal. It's almost like a relay race. Okay. The first participant is the ligand. It goes and binds to the receptor. The receptor now knows, hey, I have to perform a function. So it calls other receptors because it likes help. And once it gets those help, all those receptors together can pass on the signal to other signaling molecules downstream. And that is how the protein functions. So we thought that, okay, it functions through the kinase domain. So let's target this kinase domain and make mutations, because that's how science progresses, right? Once we can find specific targets on the protein, we could maybe use it to strategize or make drugs or inhibitors or everything. So it would be more like a clinical aspect to our research. So we targeted these specific domains which are previously known to highly be phosphorylated or autophosphorylated post ligand binding. And we made multiple. I made 25 mutants. I'm only showing you 3 mutants which are really important. One is a triple mutant, one is a pentamutant. In the triple mutant, I've specifically mutated these 3 sites. In the pentamutant, I've mutated all these 5 sites. And then I made a different mutant called a SARC mutant. Now, why did I select these 3 specific mutants? That's because the percentage EGFR activity is a direct outcome of the type of mutation. Okay, so if I make this triple mutant, there is a 50% loss of activity. If I make the pentamutant, there is only 10% activity remaining. So almost 90% of it is lost. With the SARC mutant, about 20% is lost, but a lot of pioneers in the field suggest that it's a very important mutation, so we just had it. And also what is unique about this SARC mutant is, in case of the triple and the pentamutant, the functioning or signaling is ligand dependent, whereas in case of the SARC mutation, the functioning is ligand independent. And that's just some intricate signaling, which is not extremely important for the purpose of this talk. But we made these mutations and we wanted to see if any of these mutations specifically regulate the observed tension and cell spreading phenomenon. So we went back to our silencing approach. We silenced the cells and then we rescued the phenotype with the mutation. So if I had to break down the experiment, I will first go into the cell, destroy all the internal protein it has, and then put this outside mutant protein into the cell, almost such that it will be like a clean background for me to check the effects of the mutants. And what is interesting to look at is we observe near complete rescue with the triple mutant. The triple mutant, the phenotype of tension seems to be very similar to the wild type cells. You know, the organization is similar to the wild type cells. If we put the pentamutant, the cell hates it. It looks like as if it's behaving like the cells without EGF stimulation, although there is EGF. So pentamutant cannot rescue the observed phenotype. Only the triple mutant can. And the SARC mutant does not seem to have much of an effect. It seems very similar to the triple mutant. Indeed, it looks a little affected compared to the wild type. However, it also seems to rescue back. So with this and all the quantifications, we can conclude that wild type EGFR by itself can definitely rescue the effect of EGFR silencing. That is again reiterating our claim that all the observed changes in tension and spread area are EGFR dependent. And the other claim would be it is indeed the kinase domain of EGFR which is actively involved in the process of integrin tension generation, morphology, and focal adhesion changes. And finally, the big claim is everything is ligand dependent, because if there wasn't a ligand, and we've done this experiment with the SARC mutant, there doesn't seem to be a big change. And that at least convinces us. And since the paper is accepted, I can tell you it is now convinced everyone that all these changes are ligand driven and not ligand independent. With that in mind, I'm gonna go to my model to try and re-explain every experiment I've done. I've not shown at least 15 experiments which were done for the purpose of time. So I will be more than happy to discuss and detail out anything if anyone is interested. But for the purposes of the talk, just look at this signaling pathway. So what happens in a cell? Okay, let's look at it. Let's consider this a cell membrane and consider the two proteins in question. One is integrin, one is EGFR. Okay. Under normal circumstances, if there is no ligand, then integrin will try to engage its extracellular matrix. Okay. It will try to form those bonds and those connections called focal adhesions. But there is no signaling crosstalk from EGFR to the integrin. Since there is no crosstalk, there is no spreading. The cell is not happy. There is no organization. There is no radial distribution. On the contrary, if there is the EGF ligand, while integrin continues to do its work, EGFR itself becomes a mechano organizer. It becomes a leader. It says, okay, I am going to help integrin out in performing its functions. And how does it help it? It helps bring about this cytoskeletal organization. It brings about radial distribution of the focal adhesions, and it helps polymerize the actin, which would help cell spreading. The other aspect, which I never talked about, is all these experiments were done in multiple different thresholds, and we can bring this broad conclusion, and it's important. And let me try to explain that. So let's say you have a low tension threshold, but you have extremely high EGFR signaling, then that phenotype can still be rescued. Okay. So it's not just the underlying substrate tension, or it's not the rigidity of the underlying substrate alone that is dictating the tension outcome, but the signaling from growth factors in a system that actively plays a role in the process of tension generation. When you have an extremely rigid surface or a hard substrate, then you don't require as much EGFR signaling. So it's more like a compensatory mechanism where when the cell does not have the ability to spread, EGFR puts its hand up and says, hey, I'm going to help out because I know it's important for you to spread, because if you don't spread, you're not going to grow. If you don't grow, you're not going to survive. If you don't survive, the cell is going to die. So this crosstalk plays an extremely vital role in helping cell survival and growth. So just putting these thoughts again, the take-home message is EGFR is the direct regulator of all these spatial and geometric organization changes that you see, especially when it comes to cellular tension. Also, EGFR is the protein which actually tunes the tension threshold for integrin activation, irrespective of whether the substrate is hard, soft. And the other observation to take home is the maturation and organization processes observed in the cell are a direct outcome of the crosstalk between these two proteins. And in turn, they facilitate cell spreading. Finally, if someone were to ask me what aspect of EGFR controls the outcome, it is the ligand-dependent binding. that is controlling the outcomes in terms of tension and focal adhesion organization. And the last thing would be, together in a cell, these two proteins act as a joint sensing apparatus. Um, why I like to mention it as a joint sensing apparatus is because I consider these two proteins as a rheostat. You tune the rheostat. When you feel cold, you go and bring down the temperature of the air conditioning. Likewise, these two proteins compensate for each other's activity. And in doing so, they help sense the environment. They help sense the response and changes and help a cell maintain normal cellular homeostasis. So this work got great reviews and was really successful. It was published pretty recently, but I also got an interview because of the work, and then I got the journal cover for all the work. This is like bioart. I don't know if you have been observant throughout, but I had something at the very beginning where I had something which looked like an owl. This one looks like a flower. This is something I do to like de-stress from the research. And it's too much fun to play with microscopic images. With that in mind, I would like to conclude by saying um, this is an advice I give even when people don't ask me. But if you're an undergrad, keep this in mind. If you want to go quickly, you can adopt a standalone approach, but that does not take you far scientifically. So try to collaborate and try to have an open mind. Science is not a single subject outcome. It's a multidisciplinary research. So you put in all different subjects. Like, for example, in my research, I put in chemistry, I put in mechanics, I put in biology, I put in biochemistry, microscopy. So, and it's like putting different colors together, but you will get out into a prism, and then you would get a single beam out. And it's almost like a reverse prism actually, but that's how I consider science, and that's how I encourage any young scientist to be open to collaborations with people who might not be from your field, but will somehow help you. And the second thing I like to tell everyone is seeing the full elephant, where don't be so narrow-minded or so specific where you forget the big picture. If you are so focused on the elephant and its really small parts of it, you will be blind towards what you're looking at. So keep an open mind, keep an open approach while doing science. With that in mind, I would like to thank my lab. This is Alexa. This is her baby. The baby is just a year and a half old now. And this is my lab and lab members. We regularly go out and go drinking and stuff. And my collaboration team, a lot of these people are like chemists and mechanical engineers who help me come up with the strategies, especially Khalid. He's my top collaborator. Who helped me devise the strategy and execute it. With that in mind, I would say I would thank everyone again for attending the talk, especially my family, my mom, dad, sisters, and my brother-in-law. I am absolutely sure they're watching this. And I hope the force is with you when it comes to studying and doing research. And I will be open to take any more questions if there are.
Yes, Tejeshwar, there are questions.
Yes.
Hajra and Chinmay, you can pick up the questions from the chat box.
So how do I go about? Should I go back reading through the questions, or will someone ask me?
Well, there will be someone who will read the questions.
Okay,
for you.
I appreciate that. Thank you so much. So let me stop sharing the screen, and then, yeah.
Chinmay?
It was a really wonderful talk, Dr. Tejeshwar. Thank you so much for it. We have a question from Sri Maya.
Chinmay?
Okay.
Yeah. Is everybody able to hear my voice properly?
I can. I am unsure of everyone else.
Okay.
Yeah.
Yes. So, yes, Chinmay, go ahead.
Yes. We have a question from Sri Maya. The interaction between the integrin and ECM, can it be used to reverse the brain cell connection which causes the degeneration of the cells?
So if I had to hand wave and answer that question without being an expert in the field of brain biology, I would say yes. Any process that is regulated and has a direct tension angle can be reversed if you find a mechanism to modulate that tension. Having said that, doing this, bringing about these changes in vivo within a brain is a million times more difficult than doing it in isolated cells. Everything that I showed you is with isolated cells. So although the answer is yes, I don't know how to do it.
Okay. So our next question is from Paridi Sharma. She's asking, SRC mutation is associated with the ligand-independent activation of the EGFR. So does it lead to cancer?
Yes. The reason we selected that mutant is because SRC-independent signaling has been associated with cancer. But the angle to that is SRC-independent signaling leads to FAC activation, which is focal adhesion kinase activation. And it's more an integrin-dependent pathway. So the crosstalk is a little complicated. However, SRC kinase is an active protein in a lot of cancer. So the answer is yes.
Chinmay, can we have the next question?
Okay. I'm so sorry for the network lag.
No problem.
So next question is from Shubham. EGFR is helping in cell signaling to downstream signaling and integrin. Hello, are you?
Yes, yes, we can hear you.
Yeah, I can definitely hear you.
Yeah, okay. So Shubham was asking, EGFR is helping in cell signaling to downstream signaling, and integrin helps in the cell ECM adhesion. So how they are both interconnected?
So I think I should have spent more time. It's not just the cell ECM connection that integrin is regulating, but there is an intracellular signaling. So there's a signaling which goes inside the cell where it forms that big structure called focal adhesion, and that focal adhesion becomes a signaling hub for all the signaling within the cell. So the molecules involved in that hub kind of crosstalk with EGFR signaling. So inside the cell too, there is definite cross communication.
Okay. Nikita is asking us, how can we selectively target EGFR receptors or integrins from cancer cells, as they are also present on the normal cells?
That's an excellent question. That is what we want to figure out, right? That is what everyone, that's $1 million question. If I knew the answer, I would be extremely, extremely rich. I would patent that technology. But to answer it in the most easiest way, you want to find novel molecular signature, right? The way people normally do it is try to target some protein or some site which is unique to cancer. So let's assume that the cancer cell has a mutation in EGFR or integrin, and that mutation leads to distortion or change in the protein conformation. Then I could use that changed structure of the protein as the site for drug binding or inhibitor binding, provided that change is significantly different from the wild type protein. Okay. As long as that happens, I could totally design a drug to that site. The issue is single point mutations normally do not do that. you need more than one site to be mutated. However, a lot of cancers are still single point mutation dependent. That's where it becomes more trickier. But if the cancer you're talking about, since you've not mentioned what cancer, so if the cancer you're talking about was bringing about a big conformational change, you could definitely design a drug or an inhibitor to it.
So the next question is from Nazreen. Is there a cell adhesion complex which mediates binding to a substrate that lacks enzyme control molecules for integrin binding?
Sorry, your voice broke in between. You will have to repeat it.
Okay, I'm just repeating the question. That's from Nazreen. Is there a cell adhesion complex which mediates binding to a substrate that lacks enzyme control molecules for integrin binding?
Is there an adhesion complex? I am confused about the question. Maybe the person is trying to ask if there is a complex which binds or there is like a structure which is independent of integrin. That's my understanding of the question. In terms of focal adhesions, no, all focal adhesions have integrin. In terms of intercellular connections, you remember the first a couple of slides I showed, like desmosomes or these mechanical junctions, they don't have integrin. So yes, in cells, there are junctions or at least communication points where there is no integrin present.
Okay. Could you just touch on the topic of the role of EFGR in cell docking? Just quickly, if we could have that.
Cell docking. Cell docking as in when the cell starts to attach to a substrate?
I guess that's the question.
So EGFR is a growth factor, right? And let's say I had trypsinized my cell. What is trypsinization? I am taking an attached cell and I'm totally detaching it. Okay, I'm totally detaching it. That process is brought about, or that transition is brought about by adding an enzyme called trypsin. And what it does is breaks all of the attachment points. Okay, so now that I've done that, if I wanted to attach it to a new surface, I would assume that is the cell docking question. So I would put the cells and then I would make sure that the media around it has all growth factors, all like good things, all positive reinforcements for the cell, where it could think that, oh, this matrix now is favorable for me to attach and grow. The first things that would form are focal adhesions, because if they don't form focal adhesions, they are not going to attach. Okay. Once focal adhesions are formed, the process of formation of focal adhesion and the process of faster growth is favored by growth factors. So growth factors facilitates this process of attachment and spreading. So it would favor cell docking in a feasibly positive environment.
I think there are a lot many questions. Chinmay, you could probably pick one last question and
yeah, you guys can totally email me. Let me put my email in the chat. So feel free to email me. I'm trying to find a universal way of putting it. Just 1 second. Can you see this slide up top here? And my email is tjrao@uab.edu. So that's my email. So if anyone has any questions, you can absolutely email me. But I'm more than happy to take more questions.
I think, yeah, Prachi, take over.
Thank you so much, sir. Now I would like to invite Ms. Priyanka Yadav to deliver the vote of thanks.
Good evening, everyone. I, Priyanka Yadav, on behalf of KC College, Departments of Biotechnology and Microbiology, deem it a great honor to propose a formal vote of thanks to all of us who has helped us in making this webinar such a resounding success. First of all, I would like to propose a hearty vote of thanks to our very dynamic principal, Dr. Hemlata Bagla, for awakening the passion and desire to support us as a ladder to reach the pole of success. Your constant encouragement has helped us in best possible ways. Thank you so much, ma'am, for always being there for us. I would also like to thank Dr. Sagarika Damle, DBT Star College Status Coordinator, for helping and supporting us in all our endeavors. I would also like to express my heartfelt gratitude to our distinguished speaker of the day, Dr. Tejeshwar Rao, proudly a KC alumnus, for his enriching talk and excellent coverage on how cells communicate with each other with the help of growth factor receptors and signaling cascades, and how novel DNA-based platforms help us to study these interactions. I thank him for gracing his crucial work and sharing with us his opinion and findings this present time. I would like to take this opportunity also to place my hearty thanks to the organizing team for being so proactive. Their meticulous work and dedication has made this event success. I would also like to thank the people who have worked behind the scene to make this event happen. That is our technical team for providing us all the help and support. Last but not the least, a special thanks to all our participants for being such a great audience and showing such a great response. Thanks for being here today, and I really appreciate that you took the time to be here and listen to this webinar. Thank you. One more.
Thank you very much.
Thank you, Tejeshwar.
Yes, sir.
Thanks.
Prabha ma'am, can you just, I've unmuted you. Can you just talk to us?
Nice to see Tejeshwar.
Hello, ma'am.